Hello, everyone. This is a quick video on the last part of the chapter that we're going to talk about. It's pretty small and simple. I wanted to give you guys this as supplementary for you guys to understand this for this week. Um, I will give you one more assignment that you need to do upon finishing this little concepts, and it's based on cladograms and cladistics, right? So let's get to it. So this is basically a section 2.9 on classification that reflects evolution. Okay, so we're talking about classification and relationships with certain features that organisms have, and ideally, that whole concept is that all organisms are related, right? And this basically means that they share a common ancestor. We all share a common ancestor, right? And this could be, between mammals or just all living organisms themselves. We share a common ancestor. Just think about the whole idea that every living organism has DNA. That can give you a sense that we all must have had a small, little, tiny, cell ancestor that we all came about from, right? And then over time, these little cells changed, diverged, started working together, and these incremental little changes inside populations of cells over millions of years is referred to as, or a long period of time is referred to as evolution, right? The changes in the population of a species is referred to as evolution, right? We'll get into that much later in more detail. Okay, and of course, just as I mentioned, this idea that all life came into existence from this common ancestor, multiple species can develop from a common ancestor, right? Common ancestor is a kind of relative term that I'm going to get into in a second. But one thing that I do want you to note is that the more evolutionary recent that a common ancestor is, because you can have various common ancestors depending on how far you want to go into this evolutionary line, right, or tree that I'm going to talk about in a second. So the more recent a common ancestor is, the more likely you're going to have two species that diverged from this common ancestor to have more related features. Okay, and I'm going to get into that in a second. Okay, so that idea of these relationships and having species that have features that are together, that they both share, can be shown into little phylogenetic trees, right? And I drew this kind of analogy of this little tree here, and phylogenetic trees are represented here by this tree. This an actual plant tree, but a phylogenetic tree is shown as these little branching diagrams, right? And they are composed of two main parts, right? You have the branching points, okay, and the branching points are, of course, where you have two branches that are dividing here. So, for example, this would be a branching point. Oops, sorry. Okay, this would also be a branching point, and so forth, right? Every time you have two divergence occurring, two branches splitting apart, you have a branching point. And these branching points, that's the location where you have common ancestors, right? Occurring, a common ancestor that is giving rise to two new species. Okay, the leaves that you see at the top, okay, that you see here, that's basically the species that has the current species that is existing today, right? And I drew several leaves because really that species, it's a whole population, right? But you're referring to as one specific species or one specific group, depending how relative the diagrams are, right? They could be organisms, or they could be broader taxonomic groups like reptilia or aves, but it's referring to one specific thing, right? In this case, I'm referring to species. Okay. Um, oh, sorry, I missed the branching point here, right? This would be another branching point. So these branches are analogous to these points here. Okay. Every branching area is a common ancestor, right? So you had these species, 2 and 3, for example, they had a common ancestor, right? And that common ancestor, the population of common ancestor, branched out into these two diverging species. Same for 4 and 5, right? They had a common ancestor. And these two groups, even 2 and 3 and 4 and 5, had a common ancestor that had similar features, but then they diverged depending on the environment, right? And ultimately, this is not in your book, but that first ancestor, that first oh, sorry guys, I don't know what's going on here. That first common ancestor is actually referred to as the last common ancestor. You don't need to know that for now, but our first common ancestor is referred to as the last common ancestor, which is pretty much a bacteria there, or a little simple eukaryotic cell, moreover. All right, so this is basically how you would show relationships. Now, going back to this idea that the more evolutionary recent the common ancestor is, the more related the two descending species are. What do I mean by that? If you notice here, let's say I'm describing this. I'm gonna put it in red here. This common ancestor, okay? This common ancestor here has evolved over time, right? More features. It's kind of, you're like narrowing down with features, right? Let's say this last common ancestor has a nucleus, right? And then you're evolving, you're changing, the population is growing more and more and more. And then you have this common ancestor for number 4 and 5. Let's say this is a mammal, right? This is a mammal, and we have hair, we have eyes, we have we're endotherms, okay, we're warm-blooded organisms. So this idea that the more evolutionary recent a common ancestor is, these two ramifying species will have more in common than if they were starting from that first points down at the bottom, right? So the descending species have more features shared. Both of these species are going to have all of these features, right? And so forth. So the higher you are in the branch, in the tree, the more related you're going to be, right? Between two branches. Okay, that's what this concept is that I want you guys to understand. All right. Regarding how you write diagrams, they could be written in different ways. They still convey the same information, right? They still convey the same information here. So if you see letter A, this phylogenetic tree is has the same information as letter B here. Now, different depending on where you are and what lab you're working in, or what research labs are focused on this, you can have different formations, right? But they all convey the same information here, right? If you notice, 4 and 5 are together here, 4 and 5 are together, okay? 2 and 3 are together, and so forth, okay? And here also, 4 and 5. This is a circular kind of diagram here. Okay, 2 and 3 are more together, and number 1 are more together. This circular one is used more when there's a lot of diversity occurring into from whatever frame you're kind of using. If you're using like hundreds and thousands of species, generally you put this circular one. But we're definitely not going to go into that detail there. Okay, at most we're going to be using A and B, but nevertheless, you have more information there. Okay, you have the same information there. All right, guys. So this is referred to as phylogenetic trees. All right. Now, phylogenetic trees can be organized in various ways, right? Depending on features that you use, DNA, molecular sequences, and to organize these and construct these phylogenetic trees, there's certain techniques that you can do, and one of the most famous techniques, or which is based on a couple of assumptions, is called cladistics. Okay, so when you make phylogenetic trees based on these kind of statements, they're referred to as cladograms, right? And you're using this assumption tool of cladistics, right? So this is a technique used to construct phylogenetic trees based on several assumptions that you kind of need to understand, right? The first one I basically explained to you guys. The more closely related two organisms are, the more characteristics they share, right? That's kind of obvious, kind of self-explanatory there. All right. The more closely related two organisms are, the more characteristics they have to share. That's why they're closely related, right? Even though some characteristics that are shared in a group, it doesn't necessarily mean that feature will always be present in more distantly related group members, right? So, for example, going back to this diagram here, it could go both ways. Let's say you have a feature here, let's say fur, right? These organisms have fur here, but it is possible that as you go along, this fur can modify into hair or some other structure that enables these further organisms to thrive. So once that characteristic is present, it doesn't 100% have to be present within these organisms, right? That go further down, that are further distantly related, right? The same thing applies to things that branch to organisms or species that branch out in completely different directions, right? Eventually, that characteristic will disappear, right? It doesn't always have to be present in every single detail. Okay. All right. And number 3, when there are multiple ways to that organisms could be related, the simplest explanation is most likely to be the correct one, right? There's several features that come and can be present in different groups. So you could kind of make a phylogenetic tree in several ways, but the most simplest explanation can give you the most accurate phylogenetic tree, right? I'm going to get back to this because it's I need to show you in a diagram for you to really understand what I'm referring to. All right, so we'll get back to this one. Definitely, I'm gonna come back to number 3 here. So when you have these assumptions and you make a phylogenetic tree, right? They're referred to as cladograms. Okay, so phylogenetic trees that uses these 3 assumptions are referred to as cladograms. Okay, they're referred to as cladograms, and when you're using specific species and specific groups, it's often used. You use a table that's referred to as a character matrix to get a sense of how to make these these cladograms, okay? And they look something like this, right? They're basically tables of characteristics that are used to tell you how organisms are related, right? And how features are shown and then absent or present depending on the organism that you're talking about. For example, this is on table 2.8. This is a character matrix of the plant groups that we've talked about before. All right. And here are the characteristics, right? Here are the characteristics. And the first characteristic, right, prefers photosynthesis. You generally put a negative if it's absent, if this characteristic is absent, or a positive if the characteristic is present, right? Photosynthesis, so every single plant species or plant group, of course, photosynthesizes, or else it wouldn't be in here, right? Vascular tissue. These bryophytes, these tiny mosses, they do not have vascular tissues. Only the ferns, the conifers, and the flowering plants, right? They have vascular tissue, the xylem and phloem. Okay? Going into more specific characteristics, seed production, right? Ferns do not produce seeds. They're seedless vascular plants. Only conifers and flowering plants. And seeds enclosed in an ovary. Basically, they're enclosed in a fruit there, and are only performed by flowering plants. So if you notice, as you go down this matrix, this table here, you start adding more specific characteristics, and you start kind of like filtering out groups. Okay. Having this said, as you have more complex characteristics, you can develop a phylogenetic tree using those features, right? Which is a cladogram. All right. That same idea can be, so this same table can be represented in this a manner, right? In this cladogram. Okay. You have a common ancestor as features are being introduced. Okay. They start branching out. So, for example, vascular tissue, those organisms that do not have vascular tissue are branched out into their own group. Okay, the bryophytes. As you talk about a second characteristic, these other organisms start branching out. Basically, the ferns, because they're starting to lose features that is common within the further groups, right? So they're branch out. Gymnosperms, the conifers, they don't produce the fruit. So they're just naked seeds, and they'll branch out into their own group, right? Until you have the flowering plants, which produce ovaries. Okay, so this same information can be used to produce a cladogram. Okay, now going back to this sense, you have the same information here. Okay. Take a look at this here. So I'm going to zoom out here. And the same information that's on this table is going to be presented here. All right. Now let's go back to this. Oh, perfect. I can show everything here. Let's go back to this number 3 here. So coming back, it says when there are multiple ways that organisms can be related, the simplest explanation is most likely to be correct. Look at this cladogram. And this cladogram, it shows here, okay, you have your first organism. You have bryophytes, right? They don't have vascular tissue, so they're branched out. Okay, so let's kind of refer this to this table here. All right, so they branched out. Then you have the pteridophytes. You have the ferns. Okay, they have to branch out because they don't produce seeds in ovaries, right? They only have vascular tissue, so they start branching out from these seeds and ovaries. But okay, from these ferns, okay, you have gymnosperms that are kind of evolutionarily related to these ferns. So they have to branch out to produce their seeds, right? Because you're talking about seeds here, right, in food production. So at the same time, you're having your flowering plants that can produce seeds and ovaries. So what I'm trying to mention is that when you make these cladograms, there's no necessity to have overly branching information. Like here, you have this branch that has seeds, but then you have seeds over here, right? You don't need to overcomplicate the phylogenetic system, right? The cladogram. Okay, so here's where it says that the simplest explanation is most likely to be correct. So it's best to just have, if we go back to this section here, it's best to have seeds present, evolving once in these two organisms, okay, than to say that you have two organisms that evolve seeds independently, right? So this is what the third claim stands for, right? When there are multiple ways that organisms can be related, the simplest explanation is the best, basically. And in this case, while the same information is present here, this cladogram is the most accurate, okay? Is the simplest and most accurate there. Okay, that's what number 3 is trying to refer to. All right? And so a little bit more information here. When you're talking about these branches, right, they're referred to as clades. And clades, more specifically, scientists try to place, because remember, you're talking about classification and also evolutionary history, right? So you're trying to place all these organisms as comfy as possible, as detailed as possible into one group, right? So whenever you have a branch, and usually when it's one branch here, okay, for example, this is referred to as a clade, okay? And it contains all organisms of a taxonomic group, okay? And when you have all of them that are just one group, like for example, the mammals, it's referred to as being monophyletic, means that it just has one branch, okay? Now, it is possible that you can have within one group the class Reptilia, okay, where they're quite different, actually, evolutionary wise. So they have to be separated into different groups. All right. So in this kind of clade, in these kind of branches, they're referred to as paraphyletic clades, that they have more than one, and they're similar enough to be in the same class, but they're different, that they need to be in their own branching diagrams, right? Into their own branches. So this is referred to as a paraphyletic group, right? Monophyletic, all of them fit in just one group. Class paraphyletic, you have multiple branches within a group, right? Birds being monophyletic also. Okay. I don't know why this is not working. Okay. Of course, there's no way you guys could know how this works. I need to give you enough information to analyze that, right? But usually, whenever you have branching diagrams like this, it generally would be a clade, a paraphyletic clade there. Okay, but of course, this is kind of changing regularly because of the DNA analysis and physical features that are being analyzed. So, but this is one of the more current, and when I mean changing, I'm talking like 5 years or 10 years or 20 years, right? Not that quickly. Yeah. So these are referred to as clades. Okay. One thing. So if you notice, clades and the thing about the plants are generally physical characteristics that in which clades are made, cladograms are made. But the problem is that cladistics does have some limitations, right? Unrelated organisms that have evolved similar characteristics completely independently, how would you fit them in a cladogram, right? Take, for example, this Australian mole, desert mole, and you have this placental golden mole here from Africa. Here you have Australia, here you have Africa, right? Where would you put these two? First of all, they look almost identical. They're around the same body size. They're around the same morphology. They have the same shape. They're blind. They have no external ears. They have internal mechanisms. They have claws. They have a blunted tail. Their skin is their fur is very silky. So they look almost identical, but these Australian moles, they're marsupials. We know what marsupials are, placentals. For the golden mole, they're placental organisms, so they're quite different, but they look almost identical. So this is a problem that cladistics has, that it can't really distinguish within these cases, right? There's always some exceptions there within these cases. Okay, and that's a limitation, right? You cannot. How do you separate really these situations, right? So there is some limitations on how to organize animals in this case. Okay. Good. And lastly, guys, we're almost finished here. This is the last thing here. Of course, like as I mentioned before, cladograms are being updated as new technology is being also developed, and new ways to analyze DNA and RNA and protein sequences. You get so much information, you develop a theory and how organisms are placed. But of course, theories can also be challenged. They can also be updated and so forth, right? To give you an example here, you have a cladogram A where you are basing it primarily on physical characteristics. And then you have cladogram B, the same three organisms, the types of organisms, basically monotremes, marsupials, and placentals. But in this case, you have the cladogram organized by molecular DNA sequencing, which shows that these two have more similarity with each other than each of them have with placental animals. But interestingly, right now, the one that is being most regarded as correct is cladogram A, right? Based on physical features here. Okay. Again, giving the sense of the mole, the golden mole, and the African mole, that definitely they'll be. It will seem that they are much different from the monotremes that lay the little eggs there, right? But this is always changing. Okay, like I had mentioned. All right, guys, so that's pretty much it for this last little part. Sorry it took a little bit more time than I had anticipated. I will give you, there is a little activity, basically lab number 2, that I want you guys to do also, but I'll give you much more time to work on it, maybe until Wednesday or Friday. Okay, guys, I'm going to post this on YouTube for you guys to, of course, you're watching this on YouTube. But the activity will be on Google Classroom for you guys to have, along with the link of this. If you have any questions, guys, let me know, and I can clarify some things in class or by email that whatever you guys need. Okay, guys, that's it for this little excerpt in classification there.